Perfect. Thank you very much. And thank you everyone for joining the discussion today. As I mentioned earlier, my name is Jeff Bates. I'm a marketing manager with IDEX Water. And again, really appreciate your time and your interest in the webinar. As you can see today, we will be discussing positive and negative culture controls, which as many of you know, are generally used for media checks on microbiology media in water testing laboratories. We're joined by two expert presenters who are looking forward to sharing their expertise with you. And before we dive into the content, I will mention that I encourage all of you to not hesitate to type your questions into the chat. We will get to those questions after the presentation, but please feel free to be asking them throughout the presentation. We'll start off with just a quick overview. The overall objective of the course is to identify best practices for positive and negative culture controls and to outline how updating your approach to these controls might benefit your organization. Our speakers will start by zooming out a bit and defining quality assurance and quality control. We certainly expect this to be a review for most people who work in water testing laboratories, but we want to make sure that everyone is on the same page before we dive into the specific practice of using positive and negative culture controls. The speakers will then make sure everyone is clear on what we mean by positive and negative culture controls, and they'll review the different methods and materials that are available to perform these controls. They will then discuss how updating your approach to these controls might benefit your laboratory, and as I mentioned, we'll be sure to leave time for Q&A. Before I introduce our speakers, I just wanted to note that the content of this webinar is based on a recent white paper, and we will provide you with a link to that white paper at the end of the presentation in case you would like to go into more depth on the subject. So now I will introduce the presenters. I'm very pleased to be joined by Patsy Root and Sean Dubois. Patsy has over 17 years of experience in water microbiology, water-related regulations, consensus standard development, and environmental laboratory accreditation. She works with several state legislative offices and industry organizations to develop sensible laws, regulations, and guidance to protect public health and ensure safe water. Patsy is also a longtime participant in various standards development organizations, including TNI, Standard Methods for the Examination of Water and Wastewater, and the American Society of Heating, Refrigeration, and Air Conditioning Engineers, or ASHRAE. She is the author of Microbiological Water Methods, Quality Control Measures for Federal Clean Water Act and Safe Drinking Water Act Regulatory Compliance. Sean is a senior scientist with IDEX Laboratories. He's been part of the IDEX water business for over 20 years, working in research and development, and in his current role, managing the product support group for North America. In addition, Sean manages the ISO 17025-2017 accreditation for the Water Quality Control Lab and is a participant in several standards development organizations, including Standard Methods for the Examination of Water and Wastewater. So thank you, Patsy and Sean, for joining us. I will now pass it over to Patsy to give us an introduction to today's topic.
Thank you, Jeff. I appreciate the introduction and welcome everybody. Happy to have you all online with us. Today, as Jeff pointed out, we're going to give some context and background about positive and negative culture controls. And for those of you who are already working in testing laboratories doing microbiology, it's probably going to be a review. But we also believe that it'll be useful content for anyone who doesn't regularly work in a laboratory, or maybe your background isn't necessarily microbiology, and for those who are new to the industry. And of course, you know, this information we hope will also be helpful to the data users. Essentially, it'll be good for all of us to be starting on the same page before we go into more depth on this particular topic. But we do feel that it's really important to remember the reasons why we perform these tasks, which is to make sure that the way that we are going about them will meet our overarching goals. May I have the next slide, please, Jeff? Thank you. So what are our overarching goals? And in here we can see a diagram outlining quality management system, quality assurance, and quality control. And ultimately, what we intend to achieve by utilizing this structure is high quality, defensible data, data that can be trusted by our customers and by the public. And in fact, this is really the fundamental purpose of why we do these water tests, because we are providing data that we hope can be trusted by our customers, and we do everything possible to ensure that, so that those customers can make good decisions about what they're going to do. But also we have to remember that regulatory determinations are made on these data, so therefore it is important to have the highest quality data we possibly can. And most importantly, we are in public health. So everything that we do to ensure the data is of highest quality possible helps protect the public. And so the way we go about this is a pretty well-defined and documented quality management system. And that's what you see here in the largest circle. But each one of those consecutively smaller circles has a very important role and impact on the overall quality management system that you have in place. The system actually defines the principles, the roles and responsibilities of personnel, and how your organization is actually going to produce data of this high defensible quality. Quality assurance, the second circle in, is those sets of activities and procedures that we undertake that help meet the quality management system objectives. And then the smaller circle, the quality control, is actually the critical part of the overall quality management system. It's still a small part, but for all intents and purposes, this is what helps us determine and ensure that our quality management program is meeting all of our intentions. And so these controls are all interrelated, but they do have some key differences between each one of them. So the key difference really is that quality management system is your framework that describes the entire process. Quality assurance refers to the management activities associated with ensuring quality data. And those are overarching activities that include how we organize ourselves, our organizational structure, how we define the roles, how are we going to assess if we're successfully producing quality data? How are we going to continuously improve? And what are we required to document and for whom? But quality control covers primarily technical activities, measuring the performance of a specific test, for instance. And that helps ensure that our systems are within the parameters that we set in our quality management system overall. Jeff, can I have the next slide, please? Thank you. So let's put a finer point on this. And here are some examples of specific quality assurance and quality control activities to try and give some clarity. So quality assurance involves activities that are mostly managerial. You're defining the roles of various personnel within your organization, like who's the technical manager, who's the quality manager, what are their requirements for the roles, and then defining policies and procedures that will lead to quality data. On the other hand, QC activities on the other side are things that you might be a little more familiar with, such as checking the incubator or the refrigerator or the freezer or the water bath temperatures on a periodic basis. Confirming that your reagents and your solutions that you use are sterile, because we don't want any introduction of extraneous organisms into the test. Ensuring that your equipment is calibrated, like micropipetters or balances. And of course, the subject for today's conversation, which is checking media performance with positive and negative culture controls. So I want to highlight 2 points from this slide. The QC activities, like I said, are very technical compared to quality assurance activities, but both are critically important to ensure that the lab is producing quality defensible data. And I also want to say that these are a small subset of the quality management system, but they are very, very key and important to us. So next slide, please, Jeff. So here you can see examples of positive and negative controls. Okay, so I want to give a precise definition of positive and negative culture controls before Sean takes over. So many of you are probably familiar with these codes, but we want to make sure we're all on the same page and understand the purpose behind these controls and what they're supposed to tell you. These controls are an important part of your quality system and evidence that your data are correct, of high quality, and defensible. So they're designed, both positive and negative culture controls, to verify the media performs the way that you expect it to, right? A positive control is the easier one to understand. It demonstrates that the test can actually find the target that you want, let's say E. coli. All right, so if we challenge a test method with a known positive strain of bacteria that should show up in this test, it should give us the expected results. So, for example, if you put a loop full of E. coli strain into Colert, you know that that test will turn up yellow and fluoresce, right? And if you put a coliform that's not an E. coli into Colert, you know that it would turn yellow, but not fluoresce. A negative control, you see on the other side, is a little bit more complicated because a lot of people think I can just run Colert media with sterile water. That is not a negative control. That's a blank or a sterility check. Okay, because what we need to determine with a negative control is that the test method won't find what it's not supposed to find. Okay, so a negative culture control cannot just be, putting sterile water in Colert media and incubating it and seeing that nothing happens. You have to actually use a negative culture control. So overall, that's a little bit of background behind the procedures that Sean's going to talk about next, and why performing positive and negative culture controls are so important to your overall quality management system and to the performance of the laboratory and ensuring to themselves and others that we are producing data of high defensible quality. So with that, I am going to pass it over to Sean.
Thank you, Patsy, and echo both Jeff and Patsy. Thank you for joining us here today for the webinar. So, as Patsy has said previously, I'm going to go over some of the different ways that a laboratory can perform positive and negative controls, culture controls for different media. So, the benefits of this this presentation, what we're going to discuss today, will kind of depend on what you might be already currently doing in your lab. So hopefully, kind of the stuff we're going to go over today will be very helpful for you, and you'll see some positives that you can take from it. So before we get too far in, I do want to acknowledge that there might be some folks who are joining us today that might be working in a lab that is not accredited and might not be required to do QC, but hopefully from what you can gather from Patsy's part of the presentation, doing QC on your media is very important, and it just ensures that the testing that you're doing is giving you accurate results that you can feel comfortable standing behind. All right, next slide, please. So as Patsy mentioned, performing controls is really important to have a known positive bacteria and a known negative bacteria. So if we take Colert, for example, Colert is a test that's going to tell you whether or not you have coliforms present, E. coli. So it's important to have a positive control. So you want to have an E. coli, so that way you can ensure that you're going to have color and fluorescence. A known coliform, so something that's just going to give you that yellow color, and then a non-coliform bacteria as your negative control. Pseudomonas aeruginosa is one that we would often suggest. So depending on the lab, you might be maintaining or having your bacteria for your QC testing done in different ways. One option, which is used quite a bit, is to maintain your own culture of your bacteria that you'll be using for QC. And this can be done by being stored on an agar plate or a slant. So this slide here kind of shows an example of what might be done in a lab that's maintaining their own culture. So obtaining a strain from a known culture collection, such as ATCC or NCTC, so that way you're getting a pure strain that is defined, so you'll know exactly what you're getting. And then from there, you would grow it onto a media as prescribed by that vendor. And then from there, you can use this plate or this slant as your working culture that you can use for your QC for the week or on a monthly basis. You can also take a portion of that and store it into a cryo vial or some other storage method that you might be using in your lab in order to keep this strain going for a longer period of time. So if you're using it for your working culture, basically you could just take a loopful, as shown here, and add it to your media that you are QCing, and then run directly. So if you were doing colert, take a loop, touch a colony from your target bacteria and your non-target bacteria, add that to individual vials with 100 mLs, add colert, and then incubate and verify that you're going to get the correct results, as we discussed earlier. You can also just keep on taking that bacteria from that plate and streak it onto a new plate or new slant to use for the following week or the following month, and then keep that going for a certain number of passages. And when I talk about passages, what I'm referring to is taking that bacteria and culturing it consecutively on new plates over a certain amount of time. We have to be very cautious of how many we do. We should never do more than 5 passages because the longer and the number of times we keep on doing this, the bacteria have a chance to actually get some mutations. And those mutations can impact the way that bacteria is going to interact with the different media that you're using in your lab. So it's always important to make sure that you do only up to 5 passages of bacteria. So in your lab, if you are maintaining your cultures, it's really important to have records of how often and how many passages you've done to ensure that you don't exceed 5. So kind of one final point on this, it's really kind of a important process because the bacteria are basically the main way you're going to QC your method or your media. And so in order to make sure that down the line you don't have issues and that everything is performing correctly, you want to have somebody who's trained in dealing with bacteria. So this usually means having like a microbiologist on staff or somebody who has those skill sets in order to make sure that there's no issues. Next slide, please. So another way of running controls for your test, rather than maintaining a culture in your lab, is to use kind of like a one-shot type of QC product. First I'm going to talk about is one that we're going to call controls with built-in swabs. So there will be a video in a little bit that we'll show that will kind of go through the overall process of this method. But just to kind of highlight it, you can see here that the way that these kind of work is that there's a plastic tube with a built-in swab. There's hydrating fluid, and then there is a pellet of bacteria contained inside. And so, again, you can kind of hopefully see in the picture that this swab at the bottom has a gray material that came from the bottom, which also had the hydrating fluid. And this is actually what is the bacterial culture for this product. So the idea with the swabs is it's supposed to be used as your starting culture, your starting point. So you would take the swab after you've run it, you would then streak it onto your like agar plate or agar slant, and this would be your starting culture. But we have found that there are water testing laboratories out there that are actually kind of using this as a one-time QC control, where after they've hydrated it and they have on the swab, they'll take that swab and directly add it to 100 mLs of their sterile water, and then they will run their media for QC. What we have found from those laboratories that are doing this is that they find the controls are very robust, meaning that they give the intended results in their QC, and that they feel that's a pretty innovative design, which it is. So again, we'll be showing a video a little bit later on that will kind of go through the whole step-by-step process for that. Next slide, please. So kind of covers some of the more popular ways that labs might be currently doing QC with both, maintaining their own cultures or the built-in swab. Some other things to talk about when we talk about controls. There's also a lot of companies out there that might provide controls along with PTs. So if you have a PT provider that is also providing QC, those would be really good to use. And what I mean by this is that, when we talk about a proficiency test or a PT, what you're getting is a test that sometimes has an unknown bacteria and an unknown concentration. And basically you're attempting to see how your lab is performing. You run the test, you send it to the PT provider, and they send you the results. Some of these PT providers may also provide a QC version of that product that can be run in the lab that's going to give you a known bacteria and a known concentration of that bacteria. What's nice about having controls that you're also getting from your PT is that it's a way to run the whole protocol with the same way that you're running your PT. It's kind of a practice run, as it were. So you have a chance to run it, you have a known number, you know what you're supposed to get, and then when it comes time to do your PT during the year, you're doing the same method, and so you can feel pretty confident that any results you're getting are not because of your process or the product, but that you're going to get some good results. A lot of these providers may also have a very large catalog of multiple strains, so you can find all the strains that you might be needing for your QC in your lab. And in general, these tend to be pretty straightforward and easy to use formats, but it's really important to pay attention to the strains that the product that you're going to be doing QC on. Know what strains are being suggested to be used for QC, so that way when you're looking in these large catalogs, you can hopefully find the one that you need in there. Because again, you don't want to choose something that is not going to give you the results that are expected from your test. One other note, depending on the vendor, they might provide a bulk number of tests, which might be good if you're doing a lot of tests or doing it very often, so it gives you lots of material. But if you're a lab that might be only required to do QC like once a quarter or once a month or once per lot, these bulk purchases might not fit very well with your lab, and you might be having a lot of waste. So it's always good to reevaluate like what you're purchasing versus what you actually need for your testing. Okay, next slide. All right, so those are some of the different approaches, different methods for doing some QC in the lab. So the next couple of slides I'm going to show, we'll look at kind of some of the different methods and the pros and cons that we found, and the ones that you can use for checking your IDEX reagents. We're specifically going to talk about IDEX QC, which we find is the most efficient, effective way to do this. So if you're using a different method today, if you're keeping your own cultures, or if you're using the built-in swab methods, hopefully some of this will kind of show some of the benefits, and you might be able to find that would be good for your lab from either from a efficiency standpoint, reduction amount of waste, or even cost. All right, so next slide, please. So IDEX QC. There's a lot of bullet points here. I'm not going to go through each one, but some of the important things to point out is that these kits are designed to contain the exact strains that are suggested for each of the IDEX reagents. So in the case of Kohler, you're going to get an E. coli. you're going to get a Klebsiella varicola and a Sumos aeruginosa as your negative control. So you're getting all of the different bacteria that are suggested to be used for testing, for QCing the product. Each one of them has been tested with the IDEX products to ensure that they are working correctly. They're generated in a lab that has several ISO accreditations, and the strains are coming directly from a culture collection such as ATCC or NCTC. So you know you're getting the right bacteria when you're getting the product. Each of the tests are going to contain a target value and an acceptable range that again have been determined based on IDEX related products. So you know when you're testing it, you should get the results as expected. And again, these are purposely built to match up with IDEX related products. So, okay, next slides, we're going to kind of talk about some of the benefits of using these kits. But first, I'd like to address that if you're a lab that's maintaining your own cultures, you know, some of this might not work if you're doing daily or weekly QC, just because a one-shot item would be probably cost prohibitive if you're having to use a new vial every single day. So kind of IDEX has developed a standard operating procedure, an SOP, to look at using an IDEX QC as kind of a starting culture that you can then use for testing your media on a daily or weekly basis. And we do have that available, and I know we'll kind of talk about later on, but you can always reach out to our water technical support team, and they have that SOP available for anybody who wants to take a look at it. So anybody who might be doing weekly or daily tests, it might be good to also just reach out and understand if this is actually required. Because while we always want to make sure QC and everything is being done correctly, there's a chance that we might be doing more QC than is required or actually needed. And that might be something just to understand for your lab, just to make sure that you're doing what's appropriate. And that might be able to help when it comes time to look at your costs or the efficiency of your lab. And again, kind of talked about when you're maintaining your own cultures, you want to have somebody who's highly trained or even a microbiologist in order to make sure that the cultures are being maintained correctly. And the amount of time they spend on that might be better used for other things. So if you can take away some of that QC responsibility for them, then they can focus on maybe some other tasks that they're needed on. So having a simple QC procedure that maybe removes the having to maintain cultures on site might be a way of helping with that. Okay. Next slide. So if you're here, I'm going to throw it back to Jeff so we can show the video mentioned earlier.
We'll start by demonstrating how to use IDEX QC for a media or method check. Start by taking only the vials you will be using out of the freezer. The vial needs to sit at room temperature for 15 minutes, and you can take care of other tasks during this time. Open the vial. Aseptically transfer the pellet to an appropriately labeled vessel containing 100 ml of sterile water. Swirl the sample to rehydrate the pellet. At this point, you may allow the vessel to stand for 5 minutes or swirl until the pellet is dissolved. Then invert the vessel to mix. The next step would be to empty the snap pack of reagent into this vessel. Now we will follow the equivalent process for built-in swab controls. Note that this is just one example of a built-in swab control. There are many different options available. This product needs to equilibrate to room temperature, which has been done. Crack the ampule filled with hydrating fluid. Apply some pressure until it breaks. Just crack it once so that no broken glass punctures the plastic. Hold vertically and tap it on the benchtop so the fluid flows to the bottom of this unit, where the pellet is. Pinch the bottom of the unit to crush the pellet until it is dissolved. Place the bacterial slurry in a rack. It is important to have this ready in advance. Open the vessel. Heavily saturate the built-in swab with the hydrated material, and then take the swab out and transfer it to the vessel of water. The next step would be to empty the snap pack of reagent into the vessel.
Cool. All right, so hopefully it was helpful seeing both methods being run kind of in a laboratory environment. So a couple of important things to kind of point out for those who might be using the swab controls is the amount of time. It's not much, but it is a little bit more to actually sit there and have to run the swab process. There are additional number of steps that are required in order to activate and have that sample prepared. So on that, the second point is that they're both very simple. It's not like they're complicated methods, but it's definitely less steps with the IDEX-QC method, and something that, thinking about, changes or growth in your lab when you're bringing on new analysts, having something that's very easy to train them to, that will take them very little time to start running. And of course, with that, the number of steps, the less steps that you have to do, the less opportunities for any kind of errors to happen, which could impact, again, the QC and the results of your tests. Next slide. And finally, it's always important to consider the amount of waste that's being generated with your processes. So if we look at IDEX-QC, you can kind of see that there's the box, which is the outer container that the material comes in. It is a paperboard, which is readily recyclable. And then the glass vial, of course, glass is also recyclable. So the only really waste that you're getting with the IDEX-QC method is just that little rubber stopper and the small amount of material at the top. Whereas if you look at the built-in swab method, basically all of the processes generate waste. The swab part has glass, the bacteria, and the swab, none of which are going to be recyclable, so they have to be disposed of. The foil bag that it comes in, not recyclable, and then depending on location and availability, the container that they come in might not be recyclable as well. So most of that is just generating waste, whereas with the IDEX-QC, most of it will be recyclable. Right. Next slide, please. So unfortunately, we don't have time to take a look at IDEX-QC and how it compares to all the different options out there for doing your QC on your media. But hopefully you get a sense of how materials that you're doing in your lab and how you're doing your QC and how this will all could be impacted in your lab. So another chart, lots of information, not gonna go through it, but there's a couple of things to point out, kind of looping back to what discussed before, where it's a really good idea if you can get a QC that matches the kind of PT format you're doing from the same company. And that way, again, you're getting that kind of built-in practice before you actually are running your PT. And that you can use these kits as kind of your starter to maintain your own cultures for a certain amount of time within the lab. Kind of to loop back on the PT provider part. So we partner with NSI, Laboratory Solutions. They will provide PTs in the same format as IDEX-QC. So if you were to contact your representative at IDEX to inquiry about the PTs, you can actually place orders for PTs through IDEX. We will then place that order with NSI, and then at that point we're completely hands off. NSI will then completely handle the rest of the process of both sending the samples to the lab, When the results are completed, they're sent directly to NSI, and then NSI will send the results. We are completely out of the loop at that point, but we can help with that that process. And again, it just helps if you're running the QC the same way you're running your PT, then you have that kind of built-in training step before the PT. Excuse me. And just to highlight again that if for you want to use kind of these kits as a starter for maintaining your own culture, there is an SOP that will be available. And again, you can reach out. I think we'll have a link later on, but you can also just contact Water Technical Support and they can provide that as well. So hopefully you can see with the product that we're kind of simplifying as much as possible for the laboratory and trying to give as many benefits as we can in order to be able to run QC when needed in your lab. So with that, again, thank you very much. And we have some time for questions.
Fantastic. Thank you, Sean. Thank you, Patsy. We'll open it up to questions. Lots of great questions coming in. I definitely want to get to as many of these as possible. We may not be able to get to all of the questions. So just before I dive into that, I want to highlight, this is the link for the white paper that sort of covers the same material, but is sort of a long form discussion of these items. So if that's of interest, you can go to this link here that you see on the page. There's some questions coming through now around the standard operating procedure that you mentioned, Sean. If you are interested in that, I definitely encourage you to reach out to our technical support team. But I will dive in and get to as many questions as possible. One that I wanted to make sure to get to, there were a couple around daily or weekly QC. And so the first one I want to hit head on is it required to run a negative and positive control every day with every batch? And then similarly, in what situation would you be required to run daily or even weekly QC? So I don't know, Sean or Patsy, who wants to take that. But
Hi, Patsy.
Yeah, well, I can tell you from the TNI standard volume 1 module 5 and ISO 17025, the requirement is that you run a positive, negative, and a sterility for every lot or batch of media. When I say lot, that's usually something that you purchase. When I say media, I typically mean that that's a media that you make in the laboratory.
And I'll just add that one example of a more daily requirement would be, I believe in Ohio, the guidance is to perform a positive control for drinking water laboratories every day. So that's just one example, but I think hopefully, Patsy, you would agree that, as you said, for the most part, it's by lot or quarterly or significantly less frequently.
But I, personally, if I'm in the laboratory, which I haven't been for many years, but still kind of know what's going on, I would run a positive and a negative with every batch of samples. So that personal preference.
So another question is it necessary to add the media to the sterile water before the pellet? So I think that was a question for you, Sean.
Yep. So the way that the product is designed is that you just add it first to water, sterile water, and then you'd add the reagent. But we have suggested to some customers, and some customers find it easier to do it the other way, where you have the reagent added first and then you add the pellet. So again, according to our the insert, you add it to water, but you can run it by adding it to water that already has the reagent dissolved.
Great. So a question that I think relates to if you're a lab that maintains cultures, like you showed on that slide earlier, is it okay to keep cultures in a broth instead of plates?
Yep, that is something that can be done. Again, it's the amount of time that you store that bacteria in a broth. You just need to verify that there's going to be no change over time, that the bacteria is still going to perform as intended. So as to how much that time is, I guess that'll be dependent. I don't know if there's anything from your side, Patsy, on a regular, government affairs or standard way that might say that you can only keep it for so long. But again, you just want to make sure that the bacteria is still viable and that it's still performing as expected.
Perfect. And another is, I think I'm going to use a little bit of interpretation here, but is it necessary to have, for a total coliform E. coli method, is it necessary to have a coliform bacteria separate from an E. coli bacteria? So probably a question for you, Patsy.
Yes. Yes. So if your method is supposed to find both coliforms and E. coli, you want to be able to discriminate and ensure that those bacteria that fall into the category of coliforms that are not E. coli can also be detected. So you need to have a positive control for coliforms. You need to have a coliform that is non-fluorescing, so it's not an E. coli and an E. coli.
Great. And yeah, just I guess another part of that question was related to ColAlert versus ColAssure. Sean, you're in a better position to do this, but I'll just say quickly, ColAssure starts out yellow and then turns purple in the presence of coliforms. In the case of yellow water or turbidity or those types of things. So I think really whether you're talking about ColAlert or ColAssure, having that coliform, that non-E. coli coliform, is important to confirm that the method is able to distinguish.
Yep.
So question around the using the QC in the PT, using the same format of QC and PT. Is it true that if you run the QC and PT at the same time and fall within the limits of the QC, you will probably be good to go for the PT?
So if you're running the QC and you're getting within the range, that means you're passing the range that was set for that particular batch. It's not a direct translation to PT. The way that PTs are handled is completely different. Basically, that's usually that's a collection of all the data that comes in from that PT round that kind of will determine what's a passing and fail. So the results that you would get are determined differently. But if you're running the method correctly and everything is performing as expected for your media, then you should see a passing result in your PT, hopefully.
Great. So interesting question about if the kits have CFU values or just MPN. I'm not sure which of you would prefer to.
Yep. Well, I can speak to that. The IDEX QC kits are, again, the IDEX QC is developed specifically for use with the IDEX reagents. So the values that are on this, the CFAs for IDEX QC are in MPN because they're based off of results obtained using IDEX products.
Great.
Can I just add to that?
Yeah.
Because I have the opportunity. Regulatory authorities globally, standards and especially ISO do use CFU and MPN interchangeably. So in other words, if you have one MPN, you have one CFU. That's actually how it is defined by ISO. So thank you.
Great. Well, I'm just looking at all of the great questions that we're not going to be able to get to because I want to be respectful of everyone's time. But again, I'll just say thank you so much to everyone for joining. We have those questions. We will follow up via email if we didn't get to your question today. But thank you, Sean. Thank you, Patsy. And thank you very much to WEF for hosting the webinar. So with that, I will pass I will pass it back over and we will close it up.
Perfect. Thank you all for presenting this information to us today. Thank you to WEF. Thank you to all of you attendees. And thank you to our sponsor, IDEX.